limit usefulness of the cannabinoids for treatment of pain therapy. So we've been very much intrigued for a number of years now by the mechanism behind this continued effectiveness of cannabinoids in chronic pain state, that is, their effectiveness with repeated treatment without appreciable development of tolerance. And together with Sam and Yoshi, formerly graduate student and a former postdoc in the lab, we studied the changes in the distribution of CB1 receptors after development of a unilateral peripheral neuropathy. And this is one caused by spinal nerve ligation in rats, and it causes essentially a development of these neuropathic pain symptoms on the site of the injury. And what we're able to demonstrate is that both the message, the mRNA, as well as protein for the CB1 receptors was upregulated, and its expression was increased on the site of the injury. We also used an excellent antibody generously supplied by Ken Mackey, one of the first speaker of the day, to show that the immunoreactivity for these cannabinoid receptors in the sensory neurons on the side of the injury that are lying within the dorsal root ganglia is also upregulated under these conditions. And so just to summarize a large set of studies, we concluded that the results, this upregulation of the endocannabinoid system together with its receptors as well as the endogenous cannabinoids, is consistent with the preserved analgesic effect of cannabinoids in neuropathic pain, and as a result provided the rational framework for the development of peripherally acting cannabinoid-based interventions that could then be used to treat neuropathic pain without the central nervous system side effects. Now, shortly after we published our findings, this is now in 2007, a consortium of pain investigators demonstrated, and what they did was they used selective deletions of cannabinoid receptors only in nociceptors. And what they were able to demonstrate is that cannabinoids mediate analgesia both in inflammatory as well as in neuropathic pain states, largely via activation of these peripheral CB1 receptors, whereas activation of central CB1 receptors are mainly responsible for the cognitive and the motor side effects of these cannabinoids. And so these findings again reinforce the idea that targeting the peripheral endocannabinoid system for therapeutic benefit could perhaps produce compounds that would be useful for treatment of neuropathic states without the side effects. Now, there are a number of different ways one could potentially approach this. One that was mentioned earlier is to go after the non-psychoactive receptors, that is the CB2 receptors. Others are to increase the endogenous endocannabinoid tone. But what we decided to focus on is, and the goal became to develop peripherally restricted CB1 receptor agonists that don't cross the blood-brain barrier. And so to achieve this goal, our medicinal chemists and pharmacologist colleagues, Herb Saltzman and Ronnie Mitra at the Research Triangle Institute in North Carolina, went on to modify CB1 agonists by introducing properties that are associated with peripheral restriction, both while maintaining affinity and activity at the CB1 receptors, as well as attempting to enhance or at least maintain metabolic stability of these compounds. So one of the great ideas we had was, well, if we simply increase the polarity of these particular chemicals, then this would prevent them from crossing the blood-brain barrier, and thereby we would achieve our goal. And as all many great ideas go, it failed miserably. I'm not going to dwell on the agony of the numerous failures, but instead focus on some of the successes that we had with alternative strategies for chemical synthesis. And in vitro screening of these successes revealed compounds. This is in the Indian series. This is the backbone, if you will, and these are the several R groups shown here. It revealed this Indian series analogs with essentially nanomolar binding affinities, both for CB1 receptors as well as for CB2 receptors. Importantly, they had minimal penetration in the kidney cell line model of blood-brain barrier permeability. You can see it by this very low number shown here. They had full agonist activity at the CB1 receptors, but were only partial agonists at the CB2 receptors. And they also were quite stable in plasma and had reasonable metabolic stability in the liver fractions. So together with Yatendra Mulpuri, formerly a graduate student in the lab, now postdoctoral scholar in the lab, we tested these new ligands, and this time we used a rat model of sciatic nerve entrapment, or SNE, on one side, and in this type of model, what you get is a very stable decrease in withdrawal thresholds. So this is termed that Allodynia term to mechanical stimulation. You can see here in dark dots, this is before the surgery, and this is after the surgery. You have this 40–50% reduction in withdrawal thresholds to hindpaw stimulation. And then we introduce PRNMI, one of the propyl substituted Indian analogs. You can see that at the peak effect, there's a complete suppression of these neuropathy symptoms, and of course it's reversible as the drug washes off within 24 hours. We also, similarly, a methoxy-substituted analog produced very similar effects, and you can see that vehicle injections don't do anything to relieve the mechanical allodynia. In some preliminary pharmacokinetic studies, you can see that there was a pretty good correspondence between the pharmacokinetics of PRNMI with the time course of its effects. Now, next, it was really important to determine if our new compounds had any central nervous system CNS side effects. So the tests that are classically predictive of central CB1 receptor activation include measures of catalepsy, hypothermia, motor performance, and analgesia. And just to show you an example, if you take a naive, self-respecting rodent and you put it on this on this test, and I don't understand why the video is not playing.
There we go.
Okay, great. So if you simply put a rodent on the ring, yes?
But we can, you have the clips right here, right? Do you want to show the clips?
Sure.
They're right here. Which one is it? The catalepsy?
This first one. You know what? Don't bother. Okay.
Okay.
Yeah, just let's go. What I was going to try and demonstrate is that normal rodents that are naive and not injected with any drug will come off the ring within about a second, whereas rodents that have been injecting with a brain permeant analog like HU210 or several of the other brain permeant analogs that are available, including delta-9 THC, will do similar effects. they exhibit catalepsy, so they'll stand on the ring, essentially catatonic for anywhere from 30 to 40 seconds, depending on the dose. And you can see that compared to this, one of our compounds has absolutely no effect either on the catalepsy, on the rotor rod, or in its ability to produce profound hypothermia. This is what happens with a Hebrew University derived compound, produces about 3 to 40 degrees drop in the core temperature. However, the only effect that we could see, and that's a small effect, this is an analgesic effect, which would be consistent with a peripherally restricted cannabinoid analog. Let me just. So, in addition, what we did was look at pharmacokinetics of these compounds, and we were able to demonstrate that the pharmacokinetics were also supportive of the behavioral data, that is the lack of CNS side effects. If you compare the amount of drug present in the cerebrospinal fluid, the active drug, compared to that of plasma, you find that this ratio is something on the order of 1 to 1,000. So very, very little of the active drug actually gets into the brain. And this is the basis of its peripheral cell activity. Now, cancer pain, as Dr. Strauss had already mentioned, is a very important clinical indication for analgesics. And oral cancers in particular are known to be very, very painful. And so our collaborators at New York University, headed by Brian Schmidt, have shown that in mice with oral cancer pain, these very mechanically hypersensitive mice, treatment with PRNMI produces a dramatic reversal of these Allodynia symptoms, and then in a very, very pleasant surprise, there was also a 40% reduction in the tumor volume after short-term treatment with this propyl substitute indene. If you take the oral carcinoma, same one that was used to produce the tongue tumors, and inoculate in the hind paw, you also see mechanical allodynia symptoms. The thresholds drop, and you can dose-dependently alleviate this allodynia by administration of PRNMI. This is systemic administration. Now, chemotherapy-induced neuropathy is a major dose-limiting side effect in the successful treatment of many cancers. And in rats, like in humans, 4 doses once per week of a platinum-based compound, cisplatin, produce both a marked mechanical allodynia as well as cold hypersensitivity or cold allodynia. What you can see, again, in this case, with an oral administration of PRNMI, there's a complete suppression of both the mechanical and the cold allodynia symptoms, and importantly, the relief appears to last for over 24 hours. This just shows the dose response. And how am I doing for time? Okay, so the last data slide that I was going to show you is that, and this I think is important for the basis of therapy, potential future therapy of chronic pain patients. What we did in this case was administer oral PRNMI. We did this at a half maximal effective dose, or EC50. So we orally administered PRNMI to these rodents with cisplatin-induced peripheral neuropathy. And as you can see, this daily administration continued for 2 weeks, produced continued suppression of neuropathy, symptoms, so there's very little, if any, development of tolerance. When we stop the treatment and then reinstated 5 days later, again, we get the appropriate response by this particular analog. So just to summarize what I've covered so far, I think we've succeeded in developing peripherally restricted cannabinoids. I just showed you some of the Indian series. There are several others that we have that may be of potential use. We showed that they have efficacy both against neuropathic and cancer pain symptoms. We also showed evidence for some anti-tumor effects, lack of appreciable tolerance upon chronic administration, and importantly, there's no psychotropic CNS side effects. So in terms of current as well as future studies, we're now continuing to work on determining the selectivity and possible off-target effects of these chemicals. We're pursuing a number of safety pharmacology and toxicology studies, and we're quite capable of potentially optimizing these PRCBs both for selectivity and efficacy. And all of this is in order to submit an IND application to the FDA and to begin clinical trials in patients who are in desperate need for effective chronic pain relief. So I just, in closing, wanted to acknowledge my collaborators at RTI, at Indiana University, at UCLA, NYU, as well as University of Arizona. Todd Wander, I didn't have time to go into the data, but we have very, very promising data with bone cancer models, another form of very painful cancer condition. Also wanted to acknowledge both past and current funding. I might add that getting these novel compounds into the clinic is going to take considerably more funding than we currently have. So if there's any pharma representatives or donors interested in investing who are tuned in to this, call me. And finally, I wanted to thank you all for listening. I'd be happy to take some questions.
All right, well, we made it. Not bad day. What a beautiful day to spend inside learning about cannabis and all that. So with that, we've reached the finish line, and our last speaker, I'm very excited to present. Normally, when I graduated from med school, I didn't even have a CV. So when he graduates from med school, he's going to have a very thick CV, so we're really excited about that. So without further ado, I want to introduce Jeffrey Chen. Jeffrey Chen graduates from medical school this June with a specialized degree with an MD and MBA, 2 degrees for the price of 1. He's the co-founder and co-director of our brand new UCLA Cannabinoid Affinity Group. Thanks again to support from the Brain Research Institute and Dr. Chris Evans. And for over the last 3 years, Jeff has really found a niche area to learn more about cannabis, and he's been focused on this topic of accelerating cannabis research. He is a Geffen Scholar recipient, a UCLA Anderson Fellowship recipient, a UCLA Wolfen Entrepreneurial Award recipient, a U.S. patent holder, and a published author in cancer research. So when we hear these rumors of these millennial generations being slackers and being difficult to work with, Jeff is not that. So we're really proud again to have Jeff come up here and tell us about accelerating cannabis research. And again, I want to thank you all for coming out on such a beautiful day and spending time with us. And again, as I said before, this is really the beginning and not just the end of us learning. All right, thank you. Come on up, Jeff.
Thank you, everybody, for sticking with us to the very end. And don't forget, we have some dessert outside and some more additional networking for you after this talk. And Tim's going to play the time dictator and give me the minutes that I've been displaying to all the speakers. So, all right, so today I'll be talking about how we can really accelerate cannabis research in America. And these are actually two media slides that were in late 2015. If you read the title of this Time magazine, I think it sums up a lot of what we've been talking about. The highly divisive, curiously underfunded, and strangely promising world of pot science. And so a few major takeaway points today. First being that there's an unprecedented amount of cannabis access, not just in California, not just in America, but around the world. And as I see it, it's actually one of the largest population health experiments of our generation. The second takeaway point that some other speakers have alluded to is that these Schedule I drug restrictions have really hampered academia. And this is an area where, medical marijuana has been legal in California for 20 years. We were the first state to legalize it, and there have been people who have been administering cannabis to patients and growing cannabis. And this is an area where academia has largely sat out on the sidelines. So in certain domains, I think that we have something that we can learn from industry. And the last part I want to talk about today is kind of these novel sources of funding that we might be able to tap into, as well as kind of new ways that we can study cannabis and new ways that we can use certain study materials, one of them namely being hemp, that other speakers today have alluded to. So talking a little bit about why study cannabis now? What's the urgency? Well, the urgency really is this unprecedented access. And some people talked about the number of states that have legalized. I think it's more useful to look at the percentage of Americans that now live with access to legal cannabis. So we talked about adult use. Any adult over the age of 21 can purchase cannabis, whether it's for medical or recreational purposes. So 21% of our country lives in states that have adult use cannabis laws. 62% live in states with medical-only laws. So despite federal Schedule I drug restrictions, a majority of Americans actually have access to medical marijuana through state-sanctioned programs. And lastly, 95% of Americans live in states that allow for some sort of legal cannabinoid. So the difference between the 62% and the 95% are the people who live in states that have legalized CBD. So CBD, despite being non-psychoactive, is still classified as the DEA claims that it's a Schedule I drug, and states have taken upon themselves to defy the federal government and legalize CBD. So this is just a map to reiterate what I talked about. And as you can see, a lot of the states in the South and in the Bible Belt, they haven't legalized medical cannabis, but they've actually gone ahead and legalized CBD, in part due to a lot of the pressure from parents of children with pediatric epilepsy. And the CNN Sanjay Gupta documentary did a lot to push a lot of this legislation forward. And so California really is the cannabis capital in the world. Even before Prop 64, we were the largest cannabis market in the world. So before Prop 64, roughly 1 million Americans had access to legal cannabis, Californians. So these are Californians who had physician recommendations and were allowed to purchase or grow their own cannabis. We don't have exact numbers because they weren't required to register with the state of California. Okay, so we had about, let's say, 1 million people. That's what the estimates round around. After Prop 64, it's about 27 million Californians. Anybody over the age of 21, so 27 million Californians. So that's a 2,007% increase in the amount of Californians who can have access to legal cannabis products. So again, unprecedented amounts of access, one of the largest population health experiments that our generation currently faces. And this is not just exclusive to California, and this is not just exclusive to America. We're talking about the globalization of cannabis. So these are all countries that have either legalized medical cannabis or, in some instances, recreational cannabis. And in fact, the Mexican parliament just passed their medical cannabis legislation. So soon we'll be the only country in North America that does not have national, federally legal cannabis. We see countries in Europe have legalized. Israel is really leading the forefront in a lot of the research around cannabis, and their government, the Israeli government, just approved the companies in Israel to begin exporting their medical marijuana products around the world as a way to boost their economy. And we see that Canada is actually, the Canadian medical marijuana companies are actually now, they're publicly traded on the Canadian stock exchanges, and they're also now exporting to countries around the world for clinical trials. They're exporting their medical cannabis products. So America, if we don't catch up, we're going to be left behind as other countries in the world jump on this. So let's take a step back and talk a little bit about the history of U.S. cannabis research. We don't have time to go all the way back to the Marijuana Tax Act in the 1930s. But let's start in the 1970s, which is the most recent iteration of the Controlled Substances Act. So it was an act that was passed in 1970, and what it did was it created categories, so-called schedules of drugs, and then it took each drug and put it into each category. So Schedule 1 drugs were the most dangerous, the most strictest. They were reserved for the most dangerous drugs, no currently accepted medical use, and a high potential for abuse. And actually, cannabis was temporarily placed in Schedule 1, but it wasn't a finalized placement in 1970 when this law was passed. They actually, at the time, it was President Nixon, and he actually appointed this commission, the National Commission on Marijuana and Drug Abuse. It's also known as the Schaefer Commission because it was led by a man named Schaefer. This was a multidisciplinary commission of legislators, regulators, congressmen, lawyers, scientists, law enforcement that were to come together and issue consensus guidelines on cannabis. And so what happened was 3 years later, they produced their 480-page report, and it basically called for marijuana to be decriminalized. And so this is 3 years later, okay? And so President Nixon, being the very level-headed, rational president that he was, he goes ahead and him and his administration take these recommendations of the commission that he appointed, and they go ahead and they make cannabis Schedule 1. And they lump it together along with drugs like heroin and LSD. And drugs like cocaine, opium, and methamphetamine actually ended up Schedule 2. So by definition, according to the federal government, by definition, having more medical use, less potential for abuse, and are in fact definitionally easier to research in America. And to go back to what Dr. A. Sola was saying in his very courageous talk right before lunch, he was saying that we need laws and policies that are guided by research and not by kind of archaic politics. And to give you an idea of what some of those archaic politics look like, they actually released the Nixon, there was about 300 minutes of Nixon tapes that weren't released until 2013. And in this one particular 30-second clip, you can actually see potentially why President Nixon at the time might have ignored the commission's findings and his administration where they were not supportive of cannabis. Sounds like somebody else we know. That's a president. Okay. So again, archaic politics not grounded in science, ignoring the findings of the commission that he appointed and put together. Now, I'd like to think that things have changed since then.
Marijuana. You gave an interview to the New Yorker's David Remnick, and you said that you thought smoking pot was a bad habit, but you didn't
let's watch this.
Worse for a person than drinking.
Sorry. So I'd like to say that since 1970 things have changed, but unfortunately nothing has changed. Cannabis is still a Schedule 1 drug, and we talked about some of the research barriers that are required. Essentially, you need to get a Schedule 1 drug license, which Dr. Roth was saying he has been doing cannabis research with a Schedule 1 drug license for decades. And when he went to renew it or when they went to transfer license, it still took a year. And even though he has been working with it and he's a known entity to NIDA and the DEA, it still took him a year. Even if you get the Schedule 1 drug license, your studies need to be approved by the DEA. There's special storage requirements, handling requirements. They have to do background checks on all the people involved in the lab. They have to come inspector safe, make sure nobody can drill from the floor below to steal the cannabis. And it's all this makes it very difficult. There used to be another layer of additional approval called the Public Health Service Review that needed to be done that added additional timing, but the Obama administration a year and a half ago removed that requirement. And like other researchers alluded to earlier, if you can get all of these things done and you can find the funding and get everything approved, you're still stuck with using cannabis from the University of Mississippi, which, as we all know, does not resemble cannabis on the street. It has significantly lower cannabinoid content, specifically significantly lower THC content, is essentially devoid of all other cannabinoids and is also devoid of terpenes, which we've mentioned briefly during the talks today, but these are these aromatic plant compounds that aren't specific to cannabis. They're present in all plants and other plants, but cannabis has particularly abundant and concentrated qualities of terpenes. So the NIDA has actually come under there's been a lot of calls on NIDA to stop the monopoly and allow other institutions to grow cannabis. And I'll talk about that a little bit towards the end of the slide. So one thing that some of the speakers earlier today alluded to also was hemp. So when Dr. Mueller was up here talking about how when she went to Boston with her son, she decided to carry CBD from hemp with her because arguably that's a legal source of cannabinoids, and we'll talk a little bit about the legislature. So as some of you might know, hemp used to be one of America's greatest cash crops till it was replaced by tobacco and cotton. And in fact, in the 1600s, there was actually colonial decrees that if you were a farmer, you actually had to dedicate X percentage of your crop to hemp because it was an issue of national security. They needed it for building materials, rope, fiber, and things like that. So in 2014, so hemp along with marijuana got banned. They kind of associated it with the same thing and it got banned together. In 2014, the U.S. Federal Farm Bill opened that door slightly, and it basically said hemp and marijuana are no longer the same thing. Hemp, they're both the same species, but hemp is going to be cannabis that is less than 0.3% THC, so essentially non-psychoactive. We talked about dispensary cannabis having upwards of 20-30% THC, so they define hemp as having 0.3% THC. And therefore, if you can derive these other cannabinoids from hemp, arguably it's a hemp derivative and not a cannabis derivative. And that's why you can buy CBD online. That's why a lot of these individuals and companies are comfortable going forward dealing with hemp-derived non-THC cannabinoids for precisely this law and this reason. And it also opens an interesting possibility for researchers to do research on phytocannabinoids for the first time ever without interference from the DEA. So an active area worth pursuing because it has very profound implications. Now, the other thing that's going on is that according to NIDA, there's 20 million Americans who have used cannabis in the last month. And so, and some of them are using recreationally, some of them are using medically. But there is data being generated in their consumption patterns, in the subjective effects they get from it, and the medical effects they get from it. And previously, prior to 10 years ago, there wasn't really a good way to collect this data. We might do these national telephone surveys and call people and say, hey, over the past 10 years, I want you to reflect on how much cannabis you use, and then we try to associate it with other outcomes. But the good thing is, this whole concept of crowdsourcing has now emerged, and we've crowdsourced all sorts of things. We crowdsource maps of entire cities by asking pedestrians to take out their phones and just film little video snippets, of 5-second snippets of their walk. And if you aggregate enough of these, you can actually piece together maps of an entire city. That's one small thing that has been crowdsourced. And so I think it's time, the time is ripe for us to crowdsource data on cannabis. And the first thing is the study material is now becoming standardized. So we've had medical cannabis legal in California for 20 years, and there's been no regulation, no traceability, no standardization. All of that changes with Prop 64. Starting in a year when the adult use stores open, California will have the strictest cannabis market in the country. When you buy a product, I can tell you who made it, I can tell you exactly what's in it, and the companies are going to be held to a higher standard. And so when a patient, if I'm a clinician and a patient comes to me and says they use X, someday soon I might be able to even look up the batch number of their product and know exactly where it came from, what it is, and what the cannabinoid and terpene content is in there. This has never existed before in California until Prop 64. Also, in all of our pockets now that half of you are on right now, just kidding, we have smartphones, right? These supercomputers that can collect data on our sleeping patterns and our activity. If you're a patient with Parkinson's, you can hold your smartphone in your hand and it can measure tremor amplitude over time. It can also collect subjective surveys. How's your pain? It can collect mood scales, right? How's your depression? And potentially this is an opportunity where we can collaborate with industry because they're the ones that are facing the users, and maybe we can work with them to help recruit users into our studies or to help us collect some of this data or at least distribute these surveys and these apps that we want to build so we can crowdsource a lot of this data. Because again, if I want to give you cannabis, I have to go through all these layers of DA approval. But if I'm merely studying you using your own cannabis, it's a lot easier. And we now have the technology and tools and standardized products to do so. And to give you a case example of how this has actually worked in the past. So we've talked a lot about Epidiolex today. And actually, Epidiolex was spurred because of all of the anecdotal data from the parents. So I'll give you an example. So back in 2012, GW Pharmaceuticals only had one product on the market. It was Sativex, this 50-50 oral mucosal spray of CBD and THC. They weren't even remotely thinking about servicing the epilepsy market. But after the Sanjay Gupta documentary, right around that time, they were getting a barrage of emails from parents of epileptic children. And these parents had seen other videos and they'd read forums of other parents who had given CBD to their children, and for some of them had really good results. So they started emailing GW Pharmaceuticals saying, hey, is there a clinical trial I could enroll in? I want to get a hold of some pure CBD product. And that's when GW got thinking, oh, there's something here. They kind of went, they looked at the videos, and that's when they started going down the orphan drug development pipeline. And the CEO of GW is very actually open about how it was really the anecdotal data that spurred them to pursue this route. And 4 years later, we have it finishing its phase 3 trials, pending potential FDA approval at the end of the year. And so this is not a new concept. This has been done before. But it's precisely because of these Schedule I drug restrictions that we have to resort to these kind of circumventing ways to do this kind of research. So some of you might be thinking, okay, that's great. We can do the hemp angle, we can do the crowdsourcing angle, but the real crux of it is, where is the funding going to come from? Okay, and I know that's on a lot of people's mind, and a lot of presenters have talked about it today as well. Well, so traditionally there hasn't been much funding at all for therapeutic research into cannabis. The National Institute on Drug Abuse, by definition, does not fund research into therapeutic research into controlled substances of abuse, especially Schedule I drugs, which by definition have no medical use, no currently accepted medical use. Well, one thing is the private philanthropic community is actually activated in ways that we've never seen before. So about a year and a half ago, there was a very wealthy Australian banker. He had a granddaughter who had one of these pediatric epileptiform disease conditions. He gave her CBD. She was having dozens, hundreds of seizures a day. He gave her CBD, and it really helped her. And he was so moved by this that he donated over $30 million to the University of Sydney and created their medical cannabis research center. So the donation to cannabis research for therapeutic purposes is, for the first time ever, a serious philanthropic issue. Imagine 20 years ago going to somebody and said, hey, give me $30 million to research weed, right? They would have laughed at you. Foundations over the last year or two are also supporting cannabis research in ways unimaginable. The Alzheimer's Foundation has supported research at the Salk Institute, and they've been doing some cell studies of how THC might be able to potentially treat Alzheimer's. The Lupus Foundation, the Milken Family Foundation has actually funded some of the research here looking at CBD and epilepsy, and the Michael J. Fox Foundation. Again, if you went to these foundations 10 years ago and asked them for millions of dollars to study cannabis, they would have laughed you out of the room. We're also seeing corporate philanthropy helping fund cannabis research. So Jefferson University is a university in Philadelphia, and they actually, when they launched their cannabis research program, they got over $1 million of funding from cannabis companies operating in Pennsylvania to fund their center. So again, there's another active area where that for funding for cannabis research that has never existed before. And if you look at well. And this is not showing up that well, but if you look at the mission statement for the Jefferson Center for Medical Cannabis, bullet point number 3, which isn't showing up very well, basically mentions that they want to, in addition to studying the therapeutic potential of cannabis and cannabinoids, the bullet point 3 of the mission statement was they want to foster responsible business practices amongst medical cannabis companies. So really signaling that they want to work with the industry to really move this research forward and move access forward. Now, we talked about how companies in Pennsylvania are donating to medical cannabis research, and we forget that California is the largest, by far the largest legal cannabis market on the planet, okay? Additionally, if we look at cannabis as an industry across America, it's the fastest growing industry in America. It's currently at $7 billion, and in 10 years projected to grow to $50 billion. And just to give you a sense of the scale of that, $50 billion, that's twice the size of the US wine industry and about the same size as the US smartphone industry. So if we can just peel off a tiny slice of this gigantic industry and use it towards research funding, it would be more funding than we've ever seen before, mainly at least for the therapeutic investigations into cannabis. Now, the state of California is also funding cannabis research in ways that it never has before. So with the passage of Prop 64, the state of California is going to collect tax revenue from cannabis sales, and $10 million a year starting in 2018 is going to go to public universities to study the impacts of cannabis legalization in California. Colorado did something very similar, but the Colorado research funding was pretty minimal. It was only a few million. It was a one-time gift of a few $1 million towards research, where California has this $10 million a year, every year for the next 10 years. So it's very significant. Now, for institutions that are finding that they're still strapped for cash at the end of the day, another interesting option is, I know I mentioned that the DEA recently announced that they're going to allow other organizations to grow cannabis for research purposes. And in fact, this is no small amount. If you look at the University of Mississippi, they're getting $70 million a year from NIDA to grow cannabis for research purposes. And again, they've been the sole monopoly recipient of these funds, as well as the sole grower of these funds for the last 50 years. And this is me putting on my MBA hat for a second, but whenever you have monopolies, who suffers? It's consumers. Consumers suffer because there's no competition, there's no regulation. You basically can bully people around when there's a monopoly. So this is not good, and we can see that because of this monopoly, Mississippi hasn't actually had an incentive to grow certain strains of cannabis or to really beef up its program. And so what are they doing with that $70 million? Well, I do know that here at UCLA, for nearly 100 years, we've had the botanical gardens. Okay, just saying, come follow me here. Look at these greenhouses on Tiverton. Beautiful, large greenhouses. I wonder what we could do with those. This is the community garden on North Campus at Sunset Recreation, and then the launch of the Living Amphitheater that you all should go to as we expand that presence. And then we have the medicinal herb garden. This is a real thing next to Ronald Reagan. Next time you guys walk by, you can actually see little signs showing the plants there and then the medicinal compounds contained within. So again, $70 million a year to grow cannabis for NIDA. We have the infrastructure here, something to consider if those other funding sources aren't helping us too hot. So again, some of the takeaways. There's been an unprecedented amount of access to cannabis, not just in California, but throughout the country and throughout the world. And this is one of the largest population health experiments of our generation. These Schedule I drug restrictions have hampered academia for 50 years. And so this is an area where we need to actively learn from and work with the industry, because in certain domains, they know more than we do. They've been growing it, they've been giving it to people, they've been playing around with it. There's these new sources of novel funding that we talked about. Hemp could be a potential source of legal cannabinoids that could allow researchers to do research on phytocannabinoids for the first time ever without DEA interference. And lastly, this possibility of crowdsourcing cannabis data in a relatively cheap, efficient, and scalable way to kind of recreate the phenomenon we saw, where it was these anecdotal reports of these children with epilepsy that led pharmaceutical companies and academics to really start investigating this line of work. So again, thank you very much for sticking with us to the very end today.